afternoon, everybody. I'm Jayatri, Chief Bioscientist at the Franklin Institute. Welcome back for another conversation about the science behind the COVID pandemic. This is an exciting update today because just in the past week, and in fact, 2 papers just today, were released about the vaccines currently in development to hopefully help prevent people from catching COVID-19 in the first place. As I'm sure all of you know, a vaccine is one of our best shots at bringing the pandemic under control. So everyone's been waiting with bated breath for some of these results that show us where things are in development and what we might expect in terms of a timeline moving forward. So I wanted to update you on the 3 vaccines among 23 that are actually currently in clinical trials that have actually published results of early clinical trials in peer-reviewed journals in the past 2 weeks. So before we get into the specifics of those individual vaccines, I wanted to walk you through what scientists are looking for during these early phases of testing. So let's bring up our first whiteboard drawing here. So all 3 of the vaccines that we're going to be talking about today are injected into patients. And so with any kind of new medication, the first thing that scientists look for are whether there are side effects. Obviously that's an important consideration for health and safety before we even get to thinking whether it does, has the effect that we want it to on the virus. And so how they actually track side effects is very simple. They ask patients or participants in the trial to just keep a log of how they're feeling. They'll bring them in for testing if they need to, and basically what they see is, are there any minor side effects like pain at the injection site or fever or general fatigue, as well as if there are any more serious side effects that affect the whole body. Anything that is more severe is a sign that maybe the dosage is too high. So sometimes in these early trials, they'll actually test several different dosages to see how high they can go before they start seeing these different side effects. But that's, of course, the most important thing to look at first. Then, of course, they look at whether there's an immune response in the body that's generated specific to the vaccine. And in these, before we move on, there's a question, so let's pause and answer that.
Is it true Dr. Fauci mentioned results as early as fall?
So there's a question that Dr. Fauci, Dr. Anthony Fauci, who's the head of one of the major institutes at the National Institutes of Health, who's leading the research efforts in the US, has suggested that we might see a vaccine this fall. I think that might be too early to think about widespread distribution of a vaccine, but based on where we are, and we'll talk a little bit about what we found out with these early trials, that things are progressing very quickly, and it is entirely possible that we might see the results of the next phase of large-scale trials by the fall. So that would set us up for 2021. But there's still a long way to go before we know the answers to that. So let's talk about what kind of signal scientists are looking for in terms of whether the vaccine could actually protect against the disease or not. So there are 2 branches of the immune system that scientists look at in terms of whether the vaccine is activating. One, and this is something that we've talked about before in some of our conversations, is antibodies. So antibodies are generated by one type of cell in the immune system, and these are molecules that essentially are in the bloodstream that can go throughout your body to bind and neutralize the virus itself. Okay, so that's one thing. So these are molecules that specifically go to the virus. The second piece of the puzzle are a separate branch of the immune system called T cells. And what scientists are learning are that both branches of our adaptive immune system, and we call this adaptive because they're developed specifically to the virus. But these T cells are also being activated by, potentially being activated by the virus. And what's different about the T cells is that the T cells are a special kind of cells that actually attack infected cells. So these antibodies are attacking the virus. The T cells are attacking your own cells that have already been infected. So they're kind of a backup system for each other in ways that both help you fight off and protect against the virus in the future. So they're looking at both of these signals. Having antibodies is great and is very easy to check. Having T cells as that second backup layer of long-lasting protection also happens even if you don't necessarily detect antibodies. So that's why it's important to look for both. Okay, so now let's talk about what these three trials have found out that were published in the last couple of weeks. So let's remind ourselves first about the structure of the SARS-CoV-2 virus that actually causes COVID-19 first. So it's a very small virus that has these spikes coming out of its exterior surface. And that spike protein is what forms that first attachment to the cells in your airways. So that's kind of like the red flag that's out there that your immune system looks for. So all of these viruses are using that spike protein as the target for the vaccine. But these three different vaccines use two different methods of actually presenting that spike protein to the body. So one system is what we call a viral vector vaccine. And this is where you can see I've drawn it in blue here. You take a different virus that's been modified, so it can't actually make you sick, and then you modify it to include the genetic material of the spike protein from the coronavirus. So essentially what you end up with is this other type of virus that's called an adenovirus. It's another virus that is commonly infects all of us, often with mild respiratory infections. But in this case, the vaccine is that modified adenovirus that is actually expressing the COVID coronavirus spike proteins on the surface. So both the vaccine that's been being developed by a group at Oxford University in the UK in partnership with AstraZeneca, as well as a Chinese team called CanSino Biologics, are using this type of viral vector vaccine. The other type of vaccine that's being used by a company in the US called Moderna in collaboration with the National Institutes of Health is using an mRNA vaccine. So in this case, they're kind of taking a shortcut to getting the spike protein into the body. Instead of having another virus showing off the spike proteins, they're going straight to the genes. They're going straight to the genetic material from the coronavirus, packaging it in a tiny delivery particle, and injecting that straight into the body. And they're essentially using your own body as kind of the final step in production of the vaccine, that this genetic material will integrate itself into your own cells, and your body will produce the spike protein that your immune system will then react to. So ultimately, in all of these vaccines, the spike protein is still the target. It's just a different packaging system, and the RNA vaccine relies on your own body to take that last step of producing the spike protein. Now, the reason that some of these vaccines have been so quick to advance to this stage is that scientists have been working on them for other diseases in the past. So for instance, the team at Oxford had developed this platform to develop a vaccine against MERS, the Middle East Respiratory Syndrome. So they were kind of already 5 steps ahead, at having this packaging system ready to go, and they were able to modify it for the new coronavirus very quickly. Similarly, RNA vaccines have been in development, and it's pretty simple to swap out what genetic material you're putting into the vaccine. Now, this mRNA vaccine system has never been approved for a vaccine before, so there's a few more question marks with this version than other viral vectors that have been used previously. Okay, so let's get to the bottom line. How are these working? So both the Oxford team and the CanSino team are currently publishing the results of their phase 2 trials. This means that both groups tested their vaccine and compared it to a group of participants who either got a placebo in the Chinese experiment or to patients who got a different control vaccine in the Oxford experiment. So okay, let's pause for a question before we get into the details.
There are two, actually. So first, if the virus is airborne, will we have to move all the floor stickers from 6 to 20 feet?
So that's a great question. So let's pause and pull back for a second about the idea of airborne viruses. So this is a big question right now in terms of what's the most common mechanism by which the virus is transmitted from one person to another. And we've talked a lot about transmission through what we call respiratory droplets. The particles that are released from your mouth when you talk, when you sneeze, when you breathe, when you sing. That still seems to be the most common method of transmission. The idea of airborne particles is kind of a gradient. It's a little bit fuzzy. It's really a matter of the size of the particles as to whether we're talking about the distance in which that particle can travel. So what we're generally seeing right now is suggestion that, when you're indoors, wearing masks is really important because that traps all of those particles close to you and prevents it from getting released out where it can infect a different person. Obviously, being outside where there's lots of airflow around you is the best way to reduce that risk. And it's a reason when you're inside, while a lot of buildings are looking at their ventilation systems to make sure that even inside, we're recirculating air, including outside air, to try and get rid of it, and knock down as many of those potentially airborne particles as we can. What's the second question?
Will either of these vaccines induce a T-cell response?
Okay, I'm going to get to that. So, great question, and let's talk now about what we're seeing with each of these three vaccines. So, as I mentioned, these Oxford and CanSino are both in phase 2, where they're actually comparing their vaccine in one group of participants to a second group of participants who have either gotten a placebo or have gotten a comparison vaccine. And what they're seeing is relatively mild side effects. So we do see things like pain at the injection site, some fatigue, some fever, but they tend to mostly go down, within 24 to 48 hours. So these would count as mild side effects. So that's a great sign. They also, as we go through what we see in terms of antibodies and T-cell reactions, both of these are effective in stimulating an antibody production in the body as well as a T-cell response. So that's great news. The difference between the two vaccines actually relies on what they're using as their viral vector. So remember, that's that adenovirus that's being used to kind of deliver the vaccine. And the difference between the two is that the Oxford team is using a chimpanzee virus as their delivery vector, whereas the CanSino team is using a human adenovirus vector to deliver it. Let's pause for a question before we get at why that distinction is important.
So, how effective effective is the vaccine on senior citizens? How well does this work with elderly and not just young, healthy people?
That is a fantastic question, and I'm just about to get into that, so thank you. So the first difference that we just talked about is the type of virus that they're using. And what the CanSino team found was that because they were using a human adenovirus as that delivery system, there are a lot of people who actually have already somehow, previously in their life been infected with that adenovirus. And so they already have antibodies against the packaging. So what they see is for people who already have that immunity against the package, their antibody response to the coronavirus is not as strong. So that's kind of a problem. That for those who already have that immunity, this virus is not as effective. Now, to that question about age, the CanSino team did include older adults in their study. And they found that, in addition, older adults, their immune systems were not as effective in producing antibodies. The Oxford team has only tested with relatively limited demographic of people. They tested 1,000 people, but they're all generally younger and healthy. So they don't know yet about its efficiency in older people. Let's pause for another question.
We have two, actually.
Okay.
First one, assuming any of these vaccine candidates are proven effective in the short term, what can you say about how effective they might be if the virus mutates, and is the virus mutating?
That's a great question, too. So if you remember, we talked about the fact that all of these vaccines are designed using the current sequence of the spike protein as a target. Could this change? The answer is yes, it could change. So that's one reason that having multiple vaccines, and there are other vaccines that use different mechanisms, is going to be a good thing in the long run. That being said, we're not seeing fast changes in the virus right now. Scientists have been studying the changes in the sequence over time, and so far it looks like this coronavirus mutates at about half the rate that the flu does. So that's a good that's a good sign that a vaccine is likely to be effective for a while, but it is something, of course, that we have to keep an eye on. And then the other part of that question, in terms of what are the long-term impacts of this vaccine, how long might it protect somebody, it's way too early to know that. Right now in these studies, they've only really followed the participants of this trial for a couple of months. So as we move forward, one of the things that these teams will be doing is to continue to follow the patients who are in these phase 2 trials at the same time that they start to scale up and move to larger pools of participants for testing. So to summarize these two trials, we see good response both in antibody production and in T-cell activity, but the CanSino one seems to have lesser effects in those in the antibody response, in those who already have immunity to the viral package, as well as older adults. Let's pause for another question before we talk about the Moderna vaccine.
2.
Great.
So, actually 3, one just popped up.
Okay.
Okay, so first, do the people they inject already have COVID, or are they just plain people that they decide to test
Sure.
on?
Yeah. So all of the people that go through the entirety of the study are those who have not had COVID yet. And they do this by testing them with a genetic test before they enroll. Because we want to know what their baseline levels are before the vaccine, before they've been exposed to the virus. That's a really important consideration when you're picking your participants.
Next, how has treating COVID patients changed? Is medication offered? Is there like a higher survival rate, or is the vaccine just that they're trying to use not really doing anything?
That's a great question. So let's talk about the difference between vaccines and treatments. So vaccines are intended to prevent somebody from contracting the virus in the future. Whereas different drugs and other therapies, and we'll talk about a really interesting idea of one a little bit later on, are intended to help people who are already infected by either reducing the severity of their infection or treating the symptoms. So we are seeing advances in how COVID patients are being treated. So, we're seeing things like even just turning patients over onto their stomach when they have very severe respiratory symptoms can help. So that's, doctors are learning this on the fly, and this is now becoming more widespread as doctors learn these practices and then share them. There are 3 different types of treatments that are currently in development. One is a drug called remdesivir. That's a drug that actually interferes with the virus itself replicating. So that's one option that's being tested right now. A second possibility is that, a second category of drugs rather, is that once patients have a severe immune response that results in a lot of inflammation in their lungs, because the body's immune system has kind of overreacted, then there's a second category of drugs that target the inflammation. And if you've heard of a drug called dexamethasone, that drug is in that second category to try and treat the inflammation. And then the third category is to get antibodies from people who have already recovered and inject them into patients who are currently suffering from COVID and essentially boost their immune system. We've got another question.
Okay, so for what sort of timeline are we looking at for the vaccine to finally be approved for usage? And we've got another one after that.
That's a good question. I'm going to come back to that question, so actually hold on to that. So let's get to the second question, and then we'll think about the timeline.
Have vaccines been developed for coronaviruses in the past?
That's a great question. Okay, so let's start there because that'll actually help us think about what this Moderna vaccine is doing as well. So the question is, have there been vaccines developed for other coronaviruses in the past? And the answer is that sort of that during the SARS outbreak, during the MERS outbreak, so this is the original SARS virus or the Middle East respiratory virus, that scientists started working on vaccines for them. But essentially, because it took a longer time period, the virus essentially started to die out before a vaccine was needed. And so that's where, it works in our favor now because we have this jump start on developing new vaccines. But we don't have any approved vaccines against the other coronaviruses to go off of. So we're kind of in this middle phase in terms of where we are with development. And that's where the Moderna vaccine comes in as well. That this was also a new platform, this idea of an mRNA vaccine that was being developed for other diseases. And so again, while this type of delivery system has never been approved before, it was ready to go. And to kind of wrap up the story on where the Moderna vaccine is right now, it's a little bit behind the other two. So this one has just released, published results from their phase 1 trial. So this was not a comparison study. They were only injecting it into people who knew they were getting this vaccine, just a much smaller number of people. But again, promising results here is that side effects for the lower doses were mild, so that's a good sign. And like the other ones, they do see both an antibody response and a T-cell response. So again, where we are right now is promising results from all 3 of these vaccines in the lab. Now, there's a lot of questions still about what happens next that I'll get to, but let's pause for a question.
So does the first drug you talked about for the questions, like how has COVID patients changed, medication offered higher survival rate, get rid of the virus, or does it just stop it from spreading to other people? Because you said it stopped it from duplicating.
Right, that's a great question. So inside your body, what remdesivir does is it actually interferes with the virus being able to make more copies of itself. So while it's not going to kill the existing virus in your body, it will keep the viral count down in your body. So hopefully that means that you're less likely to transmit it to other people, and that your symptoms will be less severe. Okay. What was the second question? Was there another one?
Yes. I keep seeing information that hospitals are associating and reporting COVID with non-COVID patients and deaths. It's difficult to believe the numbers.
Right, so getting data on COVID patients has been a tricky situation just because there are so many systems of reporting. I'm not personally familiar with the protocols that hospitals use to record that data, so I'm actually not going to comment on that, and that's something that maybe we can talk about in a future conversation after I learn a little bit more. But that being said, what you can see, regardless of specific protocols, is that hospitals keep track records of deaths, and actually in the general population, public health systems in cities and states keep track of how many people die every year. And so regardless of what cause that is, when you see the increases in the number of deaths in the population in general that we're seeing right now, I think that's a clear sign of the impact of the pandemic. Regardless of whether or not you can correlate any one individual's death specifically to COVID or not. So at that population level, we are absolutely seeing this impact, but I won't comment on specific data records as I'm not entirely familiar with that system. Okay, so let's talk about what happens now. So I think these vaccines give a good picture of the process by which these are going through testing. And as I mentioned, there are 20 more vaccines in development that are also going through the same process. So what happens next, and we can look at the Oxford and the CanSino vaccines in particular, is that they are now going into phase 3 clinical trials. So let's pause for a question before we get into what that means.
Can an individual receive more than one type of vaccine? For example, can one person receive both a viral vector vaccine and an mRNA vaccine? And if so, can different vaccine types be packaged together and administered simultaneously?
That is a really good question. And it really gets into what happens as we move forward. So one of the issues is production and manufacturing. And each of these has kind of a different plan for how to scale up manufacturing if and when the vaccine is approved. So while it's technically feasible for one person to get multiple vaccines, I think we'll need a lot more information before we know whether that's the most best use of our resources, if we will. Because we've got a whole world to get a vaccine to. So we'll need to understand more about the pros and cons, or whether some vaccines work better in for some type of people than others. That we'll have to think at a very kind of broad level about how to distribute the vaccine and who gets it, and whether it makes sense for to combine these vaccines to get more efficacy. We'll have to do testing as to when you combine those two vaccines, are there any unexpected outcomes? So in theory, yes, and there may be a benefit to that, especially if you're combining vaccines that attack different parts of the virus or stimulate different immune reactions. There may be a benefit to that, but I think it's too early to tell. So as these each of these vaccines go into phase 3 testing, we're looking at now tens of thousands of participants. And that's important because you want to now test the vaccine on people who have other health conditions to see how the vaccine might interact with those health conditions. You want to be able to administer this to a wider age range of people. As we discussed, the CanSino vaccine already has some indications that it's not as effective, at least for the antibody response, in older people. How does that work for kids? And then how does that work for people in different countries, people in different cities, people of different ethnic backgrounds and ancestry? These are all questions that we'll need to answer before we start manufacturing and distributing these vaccines on a large scale. So I want to wrap up with one more idea before I move on to our llama story, which is that in the U.S., if we're looking at what it means for these vaccines to get approved, the FDA has said that a vaccine needs to be effective in 50% of people in order to get approved. And so there's some discussion about whether that 50% threshold, is that too low? Should we really be looking for a vaccine that's maybe 75% effective? Or is it too high and no matter what we have, it's better than nothing, so let's get it out there. So I expect that we're going to see a lot of discussion and maybe some early emergency authorizations before we start seeing these vaccines approved for widespread use. Let's pause for a question.
I have read studies suggesting that prior infection of other coronaviruses are providing effective T-cell responses in most individuals, which is why the virus appears to just die out in certain areas. Can you comment on this?
Yeah, that's a good question, and I haven't read all of the research on that, but that is certainly a hypothesis as to why some individuals seem to be have much less severe symptoms than others. But I think we're still learning a lot about how the T-cell response is involved in the immune reaction. So I think it's a great hypothesis. I don't know that we have the data to know for sure yet. But that's actually a really good transition to the llama story. And I'm an evolutionary biologist, and so finding inspiration and solutions from other animals based on both our shared evolutionary history as well as some evolutionary tangents is something that I find really interesting. So I was really excited when I saw a story last week about a paper that has been that was published talking about how we might use antibodies from llamas and camels and other animals in that family to maybe help us find a new treatment for COVID. So I talked a little bit earlier about how among the categories of treatments that we have for COVID is to get antibodies from patients who have already recovered. Now, it turns out that llamas might be able to help us out in a similar way. So let's talk about what an antibody looks like. So in a human antibody, all right, I think we're stable here. So a human antibody is kind of complicated. So there is two heavy chains, and these are proteins. And there are two light chains. So all in all, there are 4 different proteins that come together to make one antibody. And essentially at the tips of this Y-shaped structure is what we call the variable region that essentially can make millions and millions of different versions of itself to recognize lots of different germs. Now, what's different about llamas and camels and their family is that instead of having both a heavy chain and a light chain, they only have the heavy chain. So that simplifies their immune systems right off the bat. And so when scientists discovered this means that the variable region, there's only one piece of it. You don't need both pieces from a heavy and a light chain to form that essentially it's a lock that's out there waiting for the key to be presented by the germ. So looking at this, what scientists have done now is actually just isolate just the variable region from the very tip of a llama's antibody. And because these are such tiny pieces of the protein, scientists are calling them nanobodies, because we like to call things nano when they're really, really small. And so there are three approaches that scientists are using to develop nanobodies originally derived from llamas as a potential treatment for COVID. So one group has actually already had an existing library of lots of different llama nanobodies. And what they did was they looked at all of these nanobodies in their library and picked a couple that seemed to recognize the coronavirus, and then changed the genetic sequence until they could get a really strong bond between the nanobody and the virus. So that's one approach. Another group took an approach, again, this is where some of our work on other diseases has come useful, is that there's another group that had developed nanobodies for SARS and MERS, tested them against this new coronavirus, and actually found that one of the nanobodies that they already had also recognized the new coronavirus. So that's another approach. And a third approach that obviously is going to take a little bit more time is to inject the new coronavirus into llamas and wait for them to develop a new set of antibodies that we can then turn into nanobodies. So this is just another tool in our toolkit of how we can potentially develop new treatments for COVID. So just like you can take antibodies from a recovered person and inject them into a patient to boost their immune response, it's the same idea here, except that you could potentially manufacture these nanobodies in a lab, and then you wouldn't have to get those antibodies from other people. You could just generate them as a mass-produced medicine. So that's kind of just a bonus story here about how scientists are really looking, outside the box for all of the tools that we have to treat and hopefully prevent COVID-19. So, do we have any other questions before we wrap up?
No.
Okay, well, thanks for joining me. This is an ongoing story, so we'll be back every time we've got something new to talk about. In the meantime, of course, the Franklin Institute is open now on a limited capacity basis, so come find me. I'm happy to talk about this at the museum anytime. So hopefully I will see you soon. Oh, we've got one more question.
So this is just for me.
Okay.
So you said that you will do different Facebook Lives when there's something new that shows up.
Yes.
Will that just be any day, or will it always be a Monday at 3 o'clock when there's something new? Or will it be like a Tuesday or Wednesday?
That's a great question. When can you expect me to be back? I think we'll try and keep these Mondays at 3 p.m. That's a time that we've come to expect to have these conversations, so we'll try to stick to that when we can, unless there's something that really I just can't wait for a Monday. So hopefully we'll be back here again with more conversations, and thanks again for joining us. Take care.